In this recording, we'll be discussing gametogenesis. So somatic cells are reproduced by mitosis, and their job is to produce identical daughter cells. These are body cells. These are literally every type of cell in your body except sperm and egg, which are your gametes. These are also diploid cells, so they have two full sets of chromosomes. Now, meiosis is how we make gametes. That's how we make the sperm and the egg, the sex cells. Okay? Now, fertilization occurs when we form a union, basically, between the sperm and the egg. Okay, they're going to fuse. Once they fuse, they form a zygote. Okay? The zygote will then divide via mitosis, actually, to create all of the cells necessary to grow an actual human being. Okay, so it's easy to kind of get everything mixed up in your head. So meiosis creates a sperm and an egg, but as soon as we put them together and make a zygote, we immediately need to do mitosis, because we need to take an egg, a fertilized egg, and we need to make a body. And how do we make a body? We need body cells. We need somatic cells. So we immediately switch over into mitosis. So the zygote must contain the correct number of chromosomes. So we should be getting one full set from the ovum, and we should be getting one full set from the sperm. Okay, so each of those are haploid cells. Okay, once we fuse those together, we end up with two full sets of chromosomes. That's where we get the diploid zygote from. The zygote we just mentioned will start to undergo mitosis. Once it gets going and starts to create new cells, we now call it an embryo. And then at the 8-week mark of pregnancy, it has divided enough through mitosis that we now call it a fetus. So we're going to talk about how to make sperm, and then we're going to talk about how to make eggs. Okay, so spermatogenesis. Okay, this is how we're going to make male gametes, the sperm cells. This is highly regulated by hormones from the hypothalamus and the anterior pituitary gland. Sperm production does not begin until puberty, but it will continue for the duration of the lifespan of the male that we're talking about. Okay, so we don't start until puberty, but will continue for the rest of their life. Okay, and that is going to be quite a bit different for egg production. Okay, sperm production occurs in the seminiferous tubules. Okay, so you may remember that from a previous lecture. The sperm forming cells are called spermatogenic cells. Okay, they're spermatogenic cells. It takes approximately between 64 and 72 days, which is about 10 weeks, for each spermatogonium, okay, which is basically what we start with. Okay, each of those spermatogonium in that 10-week period will produce 4 mature haploid sperm cells. Once the sperm have been ejaculated, sperm only live approximately 48 hours within the female reproductive system. Okay. Okay, so spermatogenesis, there's a few steps. Okay, step number 1, we are going to start with a spermatogonium or multiple spermatogonia. These are stem cells. Okay, this is what initiates spermatogenesis, and this is going to blow your minds. They are diploid cells. They have 2 full sets of chromosomes. They've got 46 chromosomes. I know. I've already lost you, but stick with me. Okay. These live in the basement membrane of the seminiferous tubules. Okay. So before puberty. Okay. That's key. Before puberty. Okay. Spermatogonia divide only by mitosis. Okay. Because we need to increase their numbers. Okay. We need to crank out as many of these stem cells as we can. Now, after puberty, okay, these spermatogonia become fully functional stem cells that are going to continue to divide by mitosis. Okay, but here's the key difference. Okay, after puberty, Once they divide by mitosis, one of those daughter cells is going to remain a stem cell because we don't want to run out of those or sperm production would stop. Now the second daughter cell, okay, is going to become a diploid primary spermatocyte. This primary spermatocyte is what will eventually become sperm. Okay. Now, that primary spermatocyte, still diploid, okay, we are going to push it towards the lumen of the seminiferous tubules, and it's going to undergo meiosis. So basically, it's going to split in half. This creates two secondary spermatocytes, and our one diploid cell now becomes two haploid cells. Okay, each of those secondary spermatocytes only has one set of chromosomes now. Okay. That secondary spermatocyte, both of them, will then undergo the second round of meiosis, which basically creates 4 spermatids, or spermatids if you prefer. Okay. Those 4 spermatids are destined to become fully functional sperm one day. Okay. The spermatids themselves don't quite fully look like sperm yet. They're small, they're more round. Okay, they are haploids, okay, so we're getting there. But they are non-motile. Okay. So they're definitely not ready to fertilize an egg yet. They have to undergo the next process called Spermiogenesis to become fully functional sperm. Okay, so this is as far as we've made it. We had our one stem cell, our spermatogonia. Okay, after puberty, this is going to split. Okay, this is going to split. And notice we had 2 full sets of chromosomes. We got 46, and each of these has 46. So we've done mitosis. This daughter cell, notice, stays a sperm cell, or excuse me, a stem cell. Excuse me. Okay. This right here, this loop, helps make sure that we do not run out of sperm stem cells. Okay. Now, this other daughter cell that we created by mitosis, this 46, we have got to get this down to 23. So this spermatogonium undergoes meiosis 1. Okay, here's meiosis 1. We make 2 daughter cells, and notice that we have now created haploid cells. Okay, so these are 2 haploid secondary spermatocytes. Those 2 secondary spermatocytes will then go through meiosis 2, and we get our 4 spermatids. Notice these spermatids don't look anything like sperm. Okay, so Spermiogenesis, which we're going to talk about next, is going to take our 4 immature spermatids, and they are going to mature and create 4 fully functional sperm. Okay. Okay. All right, so we got to do Spermiogenesis. This is sperm maturation. Okay, we've done all of the dividing that we're going to do. Now we're just going to grow up. We're going to change our shape and our size. Okay. As the spermatids elongate, okay, the nucleus is going to condense, and it's basically going to form a head, the little pointy end, with an acrosome at the top. Kind of looks like a little hat. Within that acrosome, you have specialized lysosomes and enzymes that help dissolve the kind of like the surrounding layer of the egg, so we can penetrate the egg to do fertilization. Okay, we get rid of any excess cytoplasm that's not really serving a purpose. The tail, which is technically a flagellum, develops, and then we have a mid-piece. It's almost kind of like a neck that contains mitochondria that are going to produce ATP to power the flagellum so we can swim. Okay, so here's how we have changed. We started as a spermatid, which doesn't really look anything like a sperm, and we start elongating. Okay, You'll notice that we got some good stuff going on at the top here as well. We're just starting to form our flagellum. This guy is the first thing that kind of, maybe a little, looks like a sperm. You can see the acrosome is forming, the tail is getting longer, and we're just continuing that process. We're making this middle piece. Okay, we've got mitochondria. We got rid of the cytoplasm that we didn't need. The acrosome is pretty well formed at this point, and by the very end we have three distinct regions: the head covered by the acrosome, the midpiece that contains the mitochondria, and the tail, the flagellum, that allows for propulsion or swimming. Now let's make an egg. Okay, so oogenesis. Okay, we're going to create an egg, an ovum, or ova is plural. Okay, but these are also haploid cells. There are some key differences between spermatogenesis and oogenesis. Okay, so first, oogenesis begins before the female infant is even born, which is kind of crazy to think about. Okay, so before the female baby is even born, we have started oogenesis, but it is suspended, so we don't finish until after puberty. Okay, now if you remember, males do not start spermatogenesis until puberty. Okay. However, once reactivated at puberty, so once we kind of pick back up where we left off at puberty, oogenesis will only continue until menopause. Okay, and we've already talked about menopause. Usually starts somewhere between 45 and about 55 years of age. Okay, so now remember, spermatogenesis, we didn't start until puberty, but we continue throughout life. Oogenesis starts before the female is born. We're going to pause until puberty. We're going to pick back up at puberty, but we're going to stop at menopause. Okay. Now, this whole process of, like, once we pick up at puberty, okay, once we reactivate this whole process, we are going to do this every month, okay, as part of the ovarian cycle to create that fully functional egg. So, the part that happens before birth, okay, we have female stem cells. They are called oogonia. Okay, they complete their mitotic division. Okay, so before birth, we take some oogonia and we do a bunch of mitosis. Okay, they undergo mitosis. We go into a bunch of mitosis. Okay, these oogonia are diploid cells. They've got their 46 chromosomes. They're created in the ovaries themselves, and they're actually going to begin meiosis 1, so the first round of meiosis, between months 3 and 7 of female development. Okay, they are going to continue to do this, and they're going to stop at basically the primary oocyte stage. Okay, so you can see we're taking an oogonium, a 2N cell, a diploid cell. Okay, we've done a bunch of mitosis to make a bunch of these. Okay, and then we're going to start meiosis. Okay, we're going to take this primary oocyte and we're going to start meiosis. We are not going to get very far in meiosis, though. We're going to do prophase 1, okay, which is like the first part of meiosis 1. Okay, this is where we're going to stop until puberty. We do this enough to create about 400,000 of these diploid cells that are paused at prophase 1. That's how many any given female has at birth. Okay, so we're doing a bunch of mitosis. We're getting about 400,000 of these diploid cells that are arrested in prophase 1. Okay, at the same time we're doing that, we have also started to create the follicle that will surround each of the eggs as well. Now, of those 400,000 diploid cells, you're not going to use very many of them. Okay, remember, puberty starts at, say 12, okay, and you stop at 45 or 50, you're only going to release about 1 egg a month for about 40 years. Okay, so you're only going to use a few hundred of your few 100,000 eggs. Okay, but we got to make sure we don't run out, right? Now, throughout childhood, nothing changes to the eggs, the primary oocyte. It just chills out. Okay. You'll notice that hormone levels cause the follicles to develop a little bit more. Okay, but the egg itself, the primary oocytes, they're not really doing a whole lot of, they're just hanging out. Okay. Now, when the hormones really start to change to trigger the ovarian cycle during puberty, that's when some fun things are going to start to happen. Okay. So, about once a month, anywhere between 20 and 30 oocytes are stimulated. Okay. To continue development. This is under the direction of secretion of follicle-stimulating hormone. Okay, so about once a month, 20 to 30 of these primary oocytes are going to be stimulated. Okay, in fact, one or more of those primary oocytes will actually complete the first meiotic division. So they're going to finish meiosis I. Okay, they're going to produce 2 haploid cells, but these are not treated equally. They differ in size. The smaller cell has DNA, but very little cytoplasm, and it's called the first polar body. It usually will finish meiosis II, but then it will just degenerate. Okay, so the little tiny cell doesn't really do anything. It eventually degenerates. The larger of the haploid cells will be referred to as the secondary oocyte. It contains the DNA. Okay, and most of the cytoplasm. Okay, this secondary oocyte has the potential to become an ovum. Okay, it will be surrounded by the Graafian follicle that we've mentioned, which has fluid in it as well. This does begin meiosis II, but it will stop again at metaphase II. Okay, the secondary oocyte will then be ovulated. Okay, and it lives for about 24 hours. If it is not fertilized within those 24 hours, it will also deteriorate and be excreted in the menstrual cycle. Okay. So you started stimulating about 20 to 30 primary oocytes. Okay. Only normally 1, maybe 2 of those primary oocytes will actually continue to develop all the way through. Okay. The larger of the haploid cells is the secondary oocyte. That is the one that is destined to become an ovum, and it has the potential to become fertilized. Okay, so we do not release 20 to 30 secondary oocytes every month. Okay, because for every one of these secondary oocytes that you release, okay, has the potential to become fertilized. Okay, and you already know that on average, the norm is single pregnancies. Okay, single pregnancies. Now, if that secondary oocyte becomes fertilized in the fallopian tube, the secondary oocyte will complete the second round of mitosis. It will do one more division and will form a second polar body. We'll get rid of that. There's no need for it. And then we finally have a fertilized ovum. Okay. Now, unlike spermatogenesis, okay, oogenesis, you end up with 3 polar bodies and 1 ovum. That's it. That's all you get. Okay. But coincidentally, it's probably all we need at one time anyway. And again, on average, right? I mean, there are triplets and, twins, quadruplets, things like that. But again, on average, the norm is just a single pregnancy. The unequal division of the cytoplasm as we continue through meiosis I and II ensures that the fertilized egg ends up with most of the nutrients, which will last them the 7-day trip to the uterus. Okay. So even though it's only a few inches long, it takes about 7 days for that fertilized egg to travel through the fallopian tubes and implant in the uterus somewhere. Okay, so here's what it looks like. Okay, we finished our primary oocyte. Okay, we are going to pick up at puberty. Okay, at puberty, we are going to split this primary oocyte. We're going to finish meiosis. We are not going to split equally, though. We're going to have one secondary oocyte that has most of the cytoplasm. This little tiny polar body, see how small it is? Okay, it just degenerates. This one secondary oocyte will kind of take another pause at metaphase 2. This is what gets ovulated. Okay, notice our follicle has grown and grown and grown. At ovulation, we pop out. Okay. Now, if we achieve fertilization of that secondary oocyte, okay, see our little sperm getting in there, we're going to do one more division. We're going to finish meiosis. Finish meiosis II. We're going to make one more polar body, get rid of that, and what we are left with is one fertilized ovum. Okay, this will go on to implant in the uterus. Our Graafian follicle that has now ruptured will degenerate into the corpus luteum, and it can start secreting the estrogens and the progesterones needed to maintain the pregnancy. All right, so just a bigger picture to put it all together. We started with the oogonium, the 2N, the diploid cells. We kind of paused at prophase 1, and then we just chilled out until we hit puberty. We picked up, we turned our primary oocyte into a secondary oocyte. Okay, if we fertilize the secondary oocyte, we finish meiosis and we create a fertilized ovum that implants and grows into a fetus.